Alright guys, I'm making this video for genetics. This will be 11.3. I'm here in 7th period. You guys want to say hi to the videos?
Hello. Hi.
Kyle, do you want to say hi to the video?
Hello.
There we go. Now you're YouTube famous. Alright, so basically this section here is about different ratios than expected with Mendelian genetics. Everything we've looked at so far has been a real easy 3-to-1 ratio, 1 and 2-to-1, things like that. And so now we're going to get in these conditions with it a little bit off from what we typically see. Not just completely dominant. In this case, it's going to be, the first one is called incomplete dominance. And an incomplete dominance, you think about it, it's not dominant, it's not the recessive, it's somewhere in between, okay? And so you see a blending of those two traits. The notes here talk about an intermediate, right? An intermediate between the two conditions, and that's what we're really talking about, an intermediate between the dominant and the recessive. One of the best examples of that you can see in snapdragons. That's a type of flower. If you breed red snapdragons with white snapdragons, you'll get all pink. Okay, now when we do that, when you do this type of reaction, you don't necessarily need, which one's dominant, which one's recessive, whether you had red or white. It doesn't really necessarily matter too much. Notice the pink is the heterozygote. Now, one thing that you'll need to notice here, though, is in these types of reactions, your genotypic ratio and the phenotypic ratio will be the same. Both, in this case, are 1 to 2-to-1. If you were to cross a pink and a pink, you'd have 1 red, 2 pink to 1 white. Your genotypic ratio would be the same: 1 big R, 1 big R, little r, and 1 little r, little r. And this just kind of shows it here. So incomplete dominance on the test, the way you can tell this is anytime I ask you a question and it says something like there's an intermediate, or it's just a physical description that's in between. A black fur mates with a white fur and you get gray. That is an example of incomplete dominance. Okay? So those things in between. Incomplete dominance is very similar to our next thing that we're going to look at, which is called codominance. Now, in codominance, two things are together. So you think about co-captains on a football team, they're equal, they're both captains. It's not a blending of those two, but it's a very specific one or the other. And so for codominance, you think about the two different alleles or two different traits. The example I would give, if you had a red flower and it mated with a white flower, in incomplete dominance, kind of looks like a smiley face, you would get some form of a pink flower. But in codominance, you'd get a flower that's red and white. And what's important to note here is that the red parts are just as red as up here, the white parts are just as white. Both traits are being equally shown. And once again, you'll have that 1 to 2-to-1 ratio for both genotypes and phenotypes. So codominance and incomplete dominance are very similar. Because it's an intermediate between the two. The biggest difference is that codominance, that there's no blending of the traits. You'll have spots. You'll have lines or streaks or parts. Okay? Incomplete dominance is a blending of the two. A good example of that is roan horse in roan color. So you take a look at this. They have what they call a red horse is up here. This is what they call a white horse. When you blend that, you get this roan color. And if you were to take a fiber from this red horse and you compared it to this one, it would be just as red. And if you compared a white hair from this to a white hair of that, it would be just as white. Both colors are being expressed equally. It's not a blending, as we saw in incomplete dominance. Another thing that can occur is called multiple alleles. So if you think about multiple alleles, instead of having just A or B, you could have a third situation. So in this case, we have blood types is a very common example of multiple alleles. And when I talk about blood typing, it's not just blood typing of A. We always hear blood typing being A, B, and O. I'm not talking about the positive and negative. That's just a simple recessive characteristic. I'm more specifically talking about the blood types A, B, and O, and how they're recessive. And so how this works is if you have type A allele, you are dominant over O. B is dominant over O, but A and B exhibit a codominance. Okay? So take a look at this, how this works out. If you are type A blood, you can be, we usually use the letter I, and because of this, we write it above. Okay? You can be this, or you can be this. Both would be type A blood. Okay? If you are type B blood, you would be this, or you would be this with the lowercase I. Now, type O blood, the only way you can get that, type O, would be if you're little I. So what's important is that when you do genetic crosses here, you can have more actual types of blood types than just are typical because of that codominance. So when we really look at this, if I were to cross someone who is, let's say A this, and I cross them with this, if I do the Punnett square, I'll just put that there, okay, oops, sorry. Filling these in. This would be type A, B blood. This is type B, drawn from here. This is type A. And then this one is little I, which is type O. So a ratio of crossing this individual with this, you'd have type AB, type B, type A, and type O. This is called multiple alleles. Multiple alleles produces more phenotypic expressions, okay? The best example of that, like I said, is blood typing. Blood typing can also be an example of codominance, considering that A and B are both equally dominant. There's not one that's dominant over the other. The next thing is something that a lot of times people ask about. They'll say something along the lines of, what about skin color or hair color? Or height. Those fall into an area called continuous variation. And the idea there is that there's not just one gene that controls height in humans. There are several. And depending upon how many of those genes are either dominant or recessive, will dramatically impact the how tall an individual is. And so we tend to see continuous variation is more shown out in a spectrum, meaning there's a lot of different varieties of height. If you have, for several genes, you could be completely recessive, you could be completely dominant, or somewhere in between. The most likely scenario would be that you're in this middle area. What this will allow for is a lot of variation between just individuals in a population or even in a family. One of the best examples I can think of is in my family. I'm 6'4", my sister's 6 foot, and my oldest sister's 5'5". We all have the same parents, all the same variation, and you could be anywhere in this spectrum, with the highest possibility being here. Intelligence exhibits continuous variation, weight, height. Okay. A lot of times when we describe these, there's a lot of variation that exists in a population. Skin color, hair color, how light or how dark your hair is, eye color, how light or how dark would all be examples. Additionally, when we talk about genetic variation, we need to account that environment influences this as well. I'll talk more about this in class on Tuesday and Wednesday, but just the idea that some genes are impacted more dramatically by the population's environment they live in, that can cause the gene to be affected by those temperatures. One of the best examples is an Arctic fox. Arctic fox have genes that are impacted by temperature, and it changes based on the season. During the summertime, they have a more of a red hue in their pigment in their fur, and during the winter, it's white. And so the genes, the expression of those genes, the phenotypic expression of those genes varies depending upon the environment. All right, so then you can kind of see these two examples here. The winter fox, okay, has its white fur influenced by the environment, and there's the summer coloration. So the environment, along with your genetics, actually factor into the expression of a particular trait. Guys, the idea is this, that when you take a look at Mendelian genetics, it's a little bit more complicated than initially thought. There's a little bit more to it. We could have cases instead of just straight dominant recessive, we can have incomplete dominance, codominance, multiple alleles, and continuous variation or environmental influence. I think it's a really good idea that you come to class with an understanding of these terms and are able to work out problems knowing these definitions. Once again, it should be just about memorizing these and then applying them. Another characteristic or important thing that we need to know about is how to determine sex based on organisms. There was a gentleman by the name of Thomas Hunt Morgan who used fruit flies as an example to help us understand sex determination. And the important thing here is that when we talk about humans, you could be an X or you could be a Y. Is what you could have. If you do a cross, that's a female, that's a male. You have a 50-50 chance of being female, 50-50 chance of being male. So that's what sex determination is. One of the things that Thomas Hunt Morgan found that was interesting was he called all the chromosomes that were not sex determination to be called autosomal chromosomes. So your first chromosome, your second chromosome, your third chromosome are all that. The X and the Y were the ones that determined sex, and initially we thought that's all they determined. So, once again, for humans, we have 23 sets of chromosomes. 22 of them are called autosomes. One set is sex chromosomes. Once again, initially, all we thought those sex chromosomes was determining the sex of the individual. But Morgan found a white-eyed male, and he crossed a white-eyed male with a normal red female. They were all red eyes. Now, if you do a simple Mendelian genetics, you think, okay, you cross red with white and you get all red. It's a dominant trait. So you cross this with this, you should get this all being red. That fits what Mendel found. Okay, so once again, being a good Mendelian scientist, we cross this with this, and what we should get is a nice 3-to-1 ratio. 3 reds to... Okay, so that's what we're hoping, or that's what he was hoping, is get 3 reds, 1 white in his F2 generation. And that's exactly what he was able to get. But the interesting point, what you really got to note here, is this line right here. All of the white-eyed flies were male. Now, based on Mendel, these two traits were unrelated to each other, were not related. You'd expect half of the white eyes to be male and half to be female, but they were all male. And so that goes away from our typical Mendelian genetic expectation, and so there has to be something else at work here. And what he realized is this important line down here, that besides just determining sex, that the X and the Y chromosome do have other traits and other genes on there. And in this case, for fruit flies, eye color was one example of that. So when we do a sex-linked inheritance, we have to do a little bit different. We have to account for X and Y. Doing that same ratio, if he had X, so that's female, big X, and we cross it with a male with white eyes, the Y does not have any characteristic on it. When you do a Punnett square like that, you would get this for the offspring. And therefore all the males would have red eyes, all the females would have red eyes in the F1. He then crossed these two, let's say right here. And when you do that, what ends up happening is you now have to keep a track for sex in the females. There's that, and there's that. Now, this would be a white-eyed male. So this is a characteristic that is found on the X and Y chromosome. You can see what this actually looks like here, the red-eyed versus the white-eyed. So sex chromosomes also have that on there. When you do a sex-linked inheritance, you need to use X and Y. Early in the year, I told you never to use X and Y, and this is the reason why, because you need them for sex-linked inheritance. This basically describes this as well. A couple characteristics are found to be sex-linked inheritance in humans. Red-green color blindness, I'll talk about in class, and hemophilia. For example, if you can't see that number in there, you might have color blindness. That's a characteristic that's more common to be, it's on the X chromosome and it's more common in males. We'll kind of talk a little bit more about that. These traits also seem to be characteristics that people say are passed from grandpa to grandson. male pattern baldness, color blindness. If you happen to be colorblind, it would be the expectation that there would be a good chance that your mother's father is also colorblind as well. And then this is just kind of going through what we did on the previous page, a little bit neater than what I did. You might want to write that down or make sure you know how to do these type of problems. Okay, continuous variation. I talked about a little bit. I want to cross that out. It's not an 8.2. A couple different examples. I'm going to pick up with this stuff in class next time. You can see the Punnett square of that for different lengths. Once again, it creates this variation that we see. That's all I have for today. I'm going to pick up on test cross, wrapping up the video. We'll pick up with test cross. These are just the notes for it, so I'm going to kind of fly through those just so you can have them. But I'll show you what a test cross is next time. That's all I got, and we'll see you in class on Tuesday and Wednesday.